Good morning, everyone. I'm Andrew Manners. This webinar will be on mite identification and management. I'm from the Queensland Department of Agriculture and Fisheries, and this webinar is part of a larger project that is funded by the Nursery and Garden Industry Levy and Horticulture and Innovation Australia. Please apologise, we've got a small lag in swapping between slides this morning. So, just as a brief overview, firstly I'm going to talk a bit about the Levy funded project. Then the bulk of the webinar is on pest mite identification and their management, and then where to get some extra information. So, this project is a building resilience and on-farm biosecurity capacity project. It's a levy-funded project, as I've said, and this webinar is only a small part of the larger project. So we're producing fact sheets, pest management plans. Part of the project is to complete diagnostics on plants that are sent in by production nurseries, and NIASA accredited nurseries get a number of free samples each year of the project, and all production nurseries get a significant discount. We're also completing education in the form of workshops and webinars, and then providing support to biosecurity through NGIA. So the information which is pertinent to you will be sent out by the NGIA newsletter and on the NGIA website. And we'll go into some detail about that later. So firstly, what we're going to do is cover what is a mite, how to identify and manage the mites, and then we'll go into some detail on pesticide usage and in conjunction with biological control. So hopefully by the end of this webinar, you'll be able to identify a rough group. These rough groups here, hopefully you should be able to look at a mite under a microscope and say, oh, I've got spider mites. Oh, I've got eriophyid mites. Okay, so firstly, what is a mite? Mites are arthropods, as are insects, crustaceans, centipedes, spiders, scorpions, and a number of other smaller groups that are less heard of. All arthropods have an exoskeleton, they have jointed legs, and they have a segmented body. And depending on the number of legs and the segmentation, the way the bodies are segmented, we group them into different categories. So mites, which is the Acari group, generally have 8 legs. Exceptions to the rule, and we'll go into that later. They have 2 body parts. They have the mouth parts, which is the first body part, and then the rest of the body. And the legs attach to that body behind in the second body part. Almost all mites are very small. If you leave out ticks, almost all mites are less than about 4 millimeters, and most of the time they're about 0.5 millimeter or less. Now, if we can make a comparison of mites to spiders, you can see that spiders also have 2 body parts, but their head end has the legs attached to it, and then the second body part, which is basically the abdomen, have very few appendages, the spinnerets where they produce silk and whatnot, but very few other appendages. So it's similar but different. The life cycle of a mite tends to go through 4 stages. So they have an egg stage, and the eggs are generally spherical or like brown, orange, red, or pink in some cases. Most of the time they're an opaque sort of a white or an off-white. In general, they hatch after a few days. The larvae, when they're present, have 6 legs. So some mites have different numbers of legs to 8. Okay, so and the larval stage, when it's present, will always have will almost always have 6 legs. So after it goes from egg, it hatches into a larva, then it molts into a nymph. And some species will have more than one nymphal stage, a maximum of 3 nymphal stages, but many mites only have 1 or 2 nymphal stages. Excuse me. Then they'll molt into an adult, and all the larvae, nymphs, and adults, they all look very similar to each other. But as the individual develops, they tend to get larger. Obviously, they gain an extra pair of legs, and they'll have more hairs on their bodies. So the adult has the most hairs on their bodies, and those are used for identification purposes. The females can lay eggs, and some species, some groups of species, don't have males at all. So the females will lay an egg. They don't even need to mate. They can lay an egg. It will turn into another female, and that female can then just start laying eggs. So the first group of mites that we're going to cover today is the largest group of mites that you're likely to see in a production nursery. They're spider mites. They're also the group which has the most commonly encountered species of spider mite, which is the 2-spotted mite. There are about 1,200 species described worldwide, but there's probably thousands, tens of thousands more than this that are undescribed. All spider mites are relatively soft-bodied. They tend to be bulbous, tend to have a bulbous-shaped body, and their mouth parts are all whip-like. So these mouth parts look basically like hypodermic needles, and because this is a squashed mite that's on a slide, it's sort of gone a bit askewed. Otherwise, those mouth parts would come straight through the middle to where the other mouth parts are, and they use those mouth parts to pierce the cell wall and suck up the cell contents. Another major characteristic of spider mites is that they produce silk, and different species of mites produce varying amounts of silk. Some will produce more, some will produce less, and sometimes the way they produce the silk is highly specialized, and we'll talk about that in a moment. So spider mites have 4 stages in their life cycle. They have an egg, which is generally spherical. They have a larval stage with 6 legs, and then they have 2 or maybe 3 nymphal stages before they turn into an adult. Males look a little bit different from females, and if you become practiced at looking at spider mites under a microscope, you'll be able to tell this difference quite readily. There are fewer males than females. If you're looking at a population, you tend to find about 25% are male and maybe 75% roughly are female. So the males tend to be a little bit narrower towards the end of the abdomen, whereas a female tends to be more bulbous. Males also tend to be more active. They're more aggressive. They'll use their mouth parts sometimes to attack other males, and they effectively fence. They're doing fencing with their mouth parts, like little tiny swords, trying to pierce the other male and kill it. Spider mites can complete their life cycle in good conditions in about 1 to 2 weeks. Sometimes it will be more, or sometimes even a little bit less, depending on environmental conditions. At low densities, spider mites tend to be present on the underside of the leaf. There are a few species which are almost always present on the upper side of the leaf, but that's relatively uncommon. At high densities, they tend to be found anywhere on a plant, and you'll have seen them just covering a plant with silk. Okay, and as we said earlier, different species produce different amounts of silk, and sometimes this can be quite specialized. So there's a species of spider mite on avocados overseas, which will produce a dense covering of silk under which they'll live and feed on the leaf. They'll have an entrance to that tent on one end, and then they'll have another covering over the top of that, and the entrance to that second tent is on the opposite side. And this sort of thing can make it quite difficult for a predator to gain access to the mites and feed upon them. Most spider mites do not have this level of complexity in their silk production, but it does occur. So I just wanted to mention it because it is an interesting sort of biological phenomenon. Spider mites tend to become most abundant during warm months, so in spring and in autumn, particularly when it's warm and dry. And this sort of condition can occur in protected cropping basically any time of the year, depending on the environmental controls of the structure. Okay, the damage that spider mites tend to cause will just vary depending on the number of mites present. So at very low numbers, you may not even see any symptoms at all, or you'll just see a very small number of chlorotic stipules, stippling. It's called stippling, where there's just the tiny little tiny yellowish spots where they've used their mouth parts to pierce the cells and simply remove the chlorophyll content of the cells. When they become more abundant, of course, this chlorosis, this stippling effect occurs over more of the leaf, and at higher densities you're more likely to have a large amount of webbing present, as you can see over here. So there will be thousands of mites on that plant. The leaves may end up having a dirty appearance, and you can see here where these little white dots, and these are the cast-off skins of molting mites. And there's also a level of silk coverage here, which makes that leaf look dirty. Sometimes the new growth can become a little bit distorted as the mites feed on the cells, causing damage, and then it can't expand that leaf properly, so there's a bit of distortion. That tends to be at the higher densities, so hopefully you don't encounter that sort of a problem very often, or at all. Now, if you've been to any of the workshops that we've been to, that John Duff and myself have been a part of from other project activities, you'll know that I'm very fond of beating plants. And what this involves is getting a tray, whether that's like the one in the picture here, or an ice cream bucket, or some other sort of laminated piece of paper. It doesn't really matter as long as it's all one color. So you beat the plant material gently but firmly against the tray, and then you can look for mites very effectively. So spider mites are very easy to find in this way. You can examine them with a hand lens then once you start to see them moving around on the plate. Other types of insects and predatory mites will also be found. With spider mites at low densities, you can beat your plants, and even though you find no symptoms, sometimes you will get spider mites be present. And then when you go back and look in more detail, you go, oh wow, there is like 1 or 2 leaves with a tiny amount of stippling and very low numbers. Now, you wouldn't necessarily have to do anything about numbers that low, but it does make it so that you can know, I need to be aware that there are mites in that area and make sure that they don't get to numbers that are high enough to cause economic damage. So plant beating is the best, as far as I'm concerned. It is one of the most efficient methods of finding spider mites in a crop. It is also worthwhile to be aware that any sort of leaf stippling, those little tiny chlorotic spots, should be a flag that you go, okay, I need to look at that plant at more detail and make sure that I know what is causing that stippling, whether it be spider mites or aphids or some other sucking insect in general. That's what it tends to be, a sucking type insect or mite. What's important to realize that spider mites populations grow very quickly. At early levels, management is relatively easy, whether that be through biological means or managing with a pesticide. If you wait until there are moderate or high levels of spider mites, it becomes more and more difficult. And we hear time after time growers spraying and spraying and just seemingly endlessly spraying for spider mites. And you want to get out of that cycle so that your plants grow better without that constant application of pesticides, and for all the other reasons as well, economics and whatnot. So it's important to record your data electronically, build patterns of pest-host associations so that you know which plant species or varieties are more likely to be hit in X particular season or under these particular conditions. That way you can devote your monitoring effort more efficiently to the high-risk crops, and they can perhaps be an indicator that you need to look in other crops for, let's say, spider mites or other pests for that matter. So the management actions that you would take against spider mites is relatively similar across a wide range of spider mite species, and it's also relevant for other mite groups. And so we'll refer back to these set of management strategies later in on this webinar. So it's always important to monitor incoming stock. The more you know about the stock that you bring in, the better. You want to make sure that it has a manageable level of pests or no pests at all, preferably. If they have pests on them, then you can take appropriate action, whether that be to return them to the supplier or to put them in quarantine and manage the problem in a smaller area as opposed to in the nursery proper. It's worthwhile reducing staff movement through areas with high infestations. Spider mites in particular are very easily brushed onto people and then onto other plants. So mites never have wings, so they can't move to different crops in that way. Excuse me. But they are very small, and their hairs can give them effectively buoyancy in the air, so they can float around on the wind quite readily, and then will at random land on plants that potentially will be suitable for them to feed on and survive and reproduce and potentially do damage. So you want to limit the amount that it moves in the crop through your own staff. So how you do that will depend on your exact nursery, but if there is some visible cue, whether it be a flag of some sort, so that a staff can avoid those high levels of infestation, then it will assist in reducing the amount that the spider might spread. Okay, so growing tolerant varieties is always beneficial. Sometimes that's not possible, and then you just have to devote your monitoring effort accordingly. Conserving natural enemies will help, because while there are a number of biological control agents available for spider mites and other mites, there are a lot more species which occur naturally in your environment and can enter the nursery and survive and have an effect on your pest species without you even doing anything. Removing weeds will help in that they're often a place where various pests can remain and then reinfest the crop. So if you remove your weeds, you're more likely to reduce your risk of any number of different pest species. And we'll discuss pesticides a little bit later. So for spider mite biological control, there are the most number of predatory mites available. Persimilis is probably the most widely used, and it's very effective. It reproduces more quickly than two-spotted mite and most other spider mites, and it moves extensively in search of prey items. So this will mean that it will eat all of the spider mites in a particular crop, and then it will move to the surrounding area in search of food. Now that's a good thing in some ways, that they're searching for food and actively going to look for it. That will mean that once the spider mites are gone, which is also a good thing, they'll go somewhere else, which is similar in a way to the application of a pesticide, which then no longer has an effect after a period of time. So Persimilis mainly consumes, it only consumes spider mites. For example, the two-spider mite bean red spider mite. If you're looking for a system to start completing biological control in your production nursery, this is the system to work with. It's the easiest to get right, and and then if you want to branch out and do more, it's a stepping stone for learning. It's always worthwhile to be in contact and build a relationship with the biocontrol agent producer. They have a lot of knowledge about their products, about their predators that I don't have, and they're specialized in. That's what they do, and they would rather have you contact them 10 times or more and get it right as you're learning how to use these predators in the crop than have no contact and have you go, oh, it doesn't work. So there are variables that we'll talk about later, and it's fairly complex. So you do need to know more when completing biological control, but it does reap rewards. So Occidentalis is a different species of predatory mite. It can withstand higher temperatures, peaks of higher temperatures and lower humidity than Persimilis, and it reproduces at a similar rate to spider mites. It can sometimes remain inactive in a crop until prey become available. So if you put it out thinking we're likely to have these spider mites, it can be inactive for a number of weeks, and then when their prey become available, they pop up and will start feeding on those spider mites. So it mainly consumes two-spotted mite, and it may feed on other mites, other spider mites and other groups of mites at a lower rate. Okay, Californicus is similar, but a different species, and similarly it will consume two-spotted mite and other spider mites at lower rates. And just as an example here, the spider mite here has that angular abdomen, which makes it a male spider mite. Now, there are various other natural enemies that will occur in your crop and will feed on spider mites. And if you see lots of spider mites and then lots of other types of mites or insects, then it is important to be aware that they could be feeding on the spider mites. For predatory mites, that teardrop shape and long legs is characteristic, and we'll see more of that as we go on in the webinar. As we said, two-spotted mite is probably the most common type of spider mite that you'll encounter. It has a very wide host range, and it also is unique in that it has a high level of pesticide resistance, or a high level of ability to develop pesticide resistance quickly. And research into the genetics of this species is quite interesting in that they show that the genome turns on different sections when they're in the presence of different types of pesticides, with different modes of actions. And that effectively allows them to tolerate or to detoxify the pesticide, particularly when they are repeatedly in the presence of that product. It has a highly variable appearance. So this is the two-spotted form, which you're more likely to see in summer or warmer periods. And sometimes you'll get this red form. And it's not just two-spotted or red. There are some shades of red and spots, if you like. So for those reasons, it is easy to blame any spider mite infestation on two-spotted mite. Superficially, it can look very similar to a number of different species which may be present in the nursery or elsewhere. And if you see, I mean, there are some differences here compared to the two-spotted mites on the previous slide, but this is also high magnification. So if you imagine you're looking at this, even with a hand lens, you go, oh, that could be two-spotted mite. We wouldn't know, even as a fairly experienced with spider mites, we wouldn't be able to tell what species you're dealing with until you get it on a slide and look at where the little hairs are, whether they're present or absent in different places on their body. Now, native plant species are likely to have native spider mites, which may well be undescribed. As we said, there are only 1,200 species of spider mites described across the world. And there are probably thousands upon thousands more than that. So your native plant species are less likely to have been studied, and it may be worthwhile having those identified, and it may assist you in being able to manage that problem. If you see unusual damage that you haven't seen before on a particular plant, it's worth having that sent for identification. From time to time, mite species which are not known to be present in Australia are found. It's relatively, I mean, it does happen every couple of years. And to my knowledge, no spider mite really has been eradicated or even attempted to be eradicated. But it can give you more information as to how to deal with that pest. Some spider mites are not readily consumed by commercial biological control agents, or may have a slightly different selection of pesticide resistance, if you know what I mean. So just knowing what you've got can help you in the management. And to that end, please send me your spider mites. I am actually completing some training in May 2017. So if in the future you can also send me spider mites, but right now particularly, I would love it if you send me your mites, send me as much foliage as you can within reason that have spider mites on them. To identify spider mites to species level, you often need a male, and as we said, there are only about a quarter of the population, so it's important to send a reasonable amount. Send it dry in a sealed bag and label it with the host plant. What I'll do is I'll be sending it, the address in an email to all the registrants of the webinar later on today. Or you can get the contact details just by Googling Grow Help Australia. Obviously, if you're not in Australia, it's important not to send samples to me. Okay, so the next group of mites we'll talk about are flat mites. Flat mites are less common than spider mites, but they can be found from time to time. They're in the same group, the same superfamily as spider mites, so they're often called false spider mites. They have a roughly similar appearance, but as the common name suggests, they are relatively flattened, and often they are orange to red in color, but not always. Their eggs are almost always that orangey red sort of color, quite bright and characteristic. Most species that you'll find do not have males. They're almost all females, and their development is a bit slower than spider mites. It tends to be about 3 to 4 weeks. Okay, they tend to be smaller than spider mites as well. So sometimes the relatively large species you can readily see with a hand lens, particularly if you have a ×20 hand lens, but not always. The very small species, you will be hard pushed, I would be hard pushed to find with a hand lens or even with a USB microscope. You really need an actual stereo microscope to see these things at about 40 times magnification with confidence. The main genera that you'll find are Brevipalpus and sometimes the Tenuipalpidae. And some of these species have been known to transmit viruses, but not very many. So orchid fleck virus is the only one that I'm aware of in Australia that can be transmitted by a particular flat mite. The damage that is caused by flat mites is somewhat varied, depending on the species and where it's feeding. So leaf stippling can occur through the same process, removing the chlorophyll from the cells, and you sometimes will get that the chlorosis of the leaves. Sometimes you'll get this russeting of the leaves, and that's fairly typical of flat mites, as well as a couple other groups that we'll talk about soon. Sometimes you'll get cupping or reduced size of the leaf, similar to broad mites, which we'll talk about soon, and sometimes you'll get premature senescence, depending on the number of mites present, etc. So if you have seeding in the new growth, you're more likely to get deformation, distortion of the stem, like in a severe case, you'll get this corkiness occurring on the stem. Sometimes this is again that russeting effect, and sometimes you'll get them underneath the leaf sheath, and that tends to cause a necrosis of that stem. But you have to be careful because a number of other species, this is in this case this is a Dianella, and you can get very similar damage caused by a Dianella gallmidge fly or from mealybugs. So it is important, this is a particular case, it's very important to identify the cause of the problem before you start applying a pesticide. Some things are relatively easy to go, oh yeah, that's an aphid, it's causing the cupping of the leaf. You can see it with your naked eye, or particularly with a hand lens. Other things, you just can't. You have to look at it under a microscope. And it's important to identify that problem because it will have different management actions, even if it is selecting a different pesticide to combat the pest. Monitoring flat mites relies on examining the plant material at high magnification. During our next workshop series coming up, starting in May and June and around the country, we will be going into some detail, John Duff and I, on how to use different tools to magnify different pests. So we'll be looking at microscopes, we'll be looking at attachments to mobile phones and whatnot. So look out for those workshop times in the NGIA newsletter and through your own state association. It's important to monitor, particularly for susceptible stock, before symptoms occur. After the symptoms occur, it's too late and you're going to have that damage for the rest of the life of the plant, unless you remove it. So I wouldn't necessarily go around looking for flat mites on every crop. It's one of those things where your records are important and you need to identify which plants you need to look at during what periods of time and to assist you in keeping your plants free of damage. So the cultural management actions are as per spider mites, and one of the problems with flat mites is that the biological control agents available commercially are not really specialized for flat mites. They might feed on them incidentally if they happen to be around at the same time as spider mites or some other prey, but they're not likely to reduce a flat mite population to below the economic threshold. So you'd be relying on naturally occurring predators, and you'd want to be keeping an eye on the numbers of predators compared to the number of the flat mites. And we'll discuss pesticides later. So the next group we'll be talking about is broad mites and cyclamen mites. These are tarsinebid mites, commonly known as thread-footed mites. It's a large family of mites. Only relatively few actually feed on plants. Most of these tarsinebid mites feed on fungi or sometimes algae. Broad mites and cyclamen mites, these are two species of mites. Even though it sort of sounds like broad mites is more than one, it's actually one species. They both have 4 life stages. They have an egg stage, a larval stage, then they have a quiescent nymph, and then it turns into an adult. To give you an idea of size, this is an immature thrips. You could just make this out with your naked eye if you're practiced at looking at thrips, and this is a broad mite. a male holding the nymphal female. We'll talk about that more in a minute. So broad mites and cyclamen mites are very small. You cannot see them without significant magnification. Now, out of interest, other tarsinemid mites have a variable life cycle. So certain species may lay live young, so they don't have eggs. Some species skip the nymphal stage, so they go from egg, larva to adult. And some species even lay reproductive individuals, so effectively they're laying adults. So that's just out of interest, that their biology is complex and interesting. Both broad mites and cyclamen mites, getting back to the real topic, have a broad host range, but broad mites are more likely to be encountered. Their damage is similar. They tend to feed in the growing tip, and they will tend to cause that cupping or deformation, shortening, causing distortion of the new growth. So cyclamen mites are more likely to occur only in the concentrated in the growing tips, whereas broad mites are still concentrated in the growing tips, but more likely to be present on some of the lower leaves. So the early symptoms would just be a slight distortion, and the later symptoms would be more severe distortion, shortening of the internodal length, petioles, more severe deformed leaves, and sometimes the plants might just appear wilted. Sometimes you will get unusual sort of symptoms with the production of anthocyanins in the leaf, such as a red pigmentation associated with stress. There's a more typical sort of infestation here where you just get deformation and dechlorosis of the leaves. More severe, then you'll have leaves falling off, not many leaves in the growing tip, and sometimes you'll just get that russeting sort of an effect on certain crops. As we said, both species are very small. Broad mites are very easy to identify. They're one of the very few species that you can identify, that you will even be able to identify just using a microscope, a stereo microscope. They're the only species which is known to have white polka dotted eggs. So this is an unhatched egg. It's got those, effectively they're bumps, but they look like polka dots. You can tell the difference between the hatched eggs and the unhatched eggs and the hatched eggs. The hatched eggs tends to be just collapsed, whereas the unhatched eggs are full. Okay, so here we have a larva, 6 legs. Then what will happen is it will turn into a nymph, a quiescent nymph. So a quiescent nymph means that it just sits around and does nothing. However, males will pick up the quiescent nymphal stage of the female, and they have a little sucker on their back and they carry it around. They wait for the female to molt into the adult stage, then they mate and take off. Okay, and as you can see here, the female is more disc-shaped compared to the male, which is more angular. So another interesting thing about mites. Okay, so cyclamen mites are similar in appearance. At least the females are similar in appearance. They're less disc-like. They're more elongated. They're slower in their movement than broad mites. And their eggs do not have polka dots. They're more typically egg-shaped. They're also less commonly encountered. Okay, so monitoring of broad mites and cyclamen mites relies on examining plant material at high magnification, just like flat mites. And you need to examine the susceptible stock before symptoms occur. Or if you start to see the slightest symptoms, then be on top of it and keep on, and then put in place management actions so that the populations don't grow. There are a number of biocontrol agents that will manage broad mites if the populations are low. But in many cases, what you'll need to do if you have a moderate, or particularly if you have a high level of infestation, you either have to get rid of some plant material, whether that be the growing tips or the whole plant. And then perhaps apply a pesticide, and then you can put on some predators, assuming you've applied the right type of pesticide. And this will then keep your numbers down. Broad mites tend to be difficult to manage with solely with pesticides. They can become quite tolerant relatively quickly. So the predators can work very effectively, but only if the populations are low. Excuse me. Thanks for that, sorry. Right, so we're now on to eriophyid mites, and we'll also talk about some naturally occurring predators. So eriophyid mites are minuscule. They're the smallest mite that you're likely to encounter in a production nursery. They have 4 life stages. You've got eggs, which you can sort of just make out here, and you've got, it's impossible to say, this is probably a larva, and maybe this one's a nymph, and the larger ones are more likely to be an adult. To give you an idea of the size of these mites, this is a spider mite that you could see with your naked eye, if you have good eyesight. Here are eriophyid mites. So it's a small proportion of the size of a spider mite. So they are very difficult to see with a hand lens unless it's at least times 20. And even then, unless you have high numbers and you're practiced at looking for them, you may have difficulties. You're much better off looking with a devoted microscope to find mites, eriophyid mites in particular. What's unique, or one of the things that's unique about eriophyid mites is they always only have 4 legs. So 2 pairs of legs, not 4 pairs of legs. So you can see them here, 1, 2, 3, 4. They're worm-like in shape or carrot-shaped, sort of broadly sort of conical. And they have many common names, including rust mites, blister mites, gall mites, erinose mites, just depending on the type of damage that they cause. And to give some examples of the types of damage, we can see here, these are called erinea. These erinea are basically hairs. The mite injects its saliva into the plant. It causes the plant to produce these finger-like, hair-like projections, and the mites live in those structures. Eriophyid mites are almost always specific to one or a small number of plant species, and they almost always, if not always, produce a very specific type of damage. So they're one of the few groups of mites where you could go, I've got that erinea or that gall on that particular host plant, I know I have eriophyid mites. You know, have a reasonable idea of the species of mite that you are dealing with. Now, when you have eriophyid mite feeding in the growing tip, then you're likely to have this sort of damage, like for the tomatoes. You might have deformation, some brown discoloration of the stems, and you know, severe cupping of the leaves. And this is the sort of thing that you'd be seeing under high magnification. These green and red galls are typical of eriophyid mites. This is the sort of thing, and there are various things. They can be slightly different shapes, and different species have, you know, slightly different galls, depending on the host plant and just the species that you're dealing with. And sometimes these galls can actually be quite pretty. Like the other spore mites that we've talked about, monitoring does rely on examining the plant material at high magnification. And if you want to prevent damage, you need to do it before symptoms occur. So again, it's one of those cases where you're looking at those highly susceptible species on a regular basis at those high risk periods of time. Once you have those eriophyid mites and the damage is present, you'll have to remove it or live with it. So on certain species, particularly if you're dealing with native plant species and a native eriophyid mite, you're always going to end up having those symptoms from time to time, whether it be you or the person who ends up having that plant in the garden or whatnot. So you have to think about whether or not it's worth trying to eradicate these things when they're going to be in the natural environment anyway. As far as I'm concerned, maybe it's just me because I like mites and whatnot, but I kind of think that these sorts of structures are funky and good. But not everyone's that way inclined, and you may decide that it is required to remove these. Certainly, if you're exporting your plants, you will need to make sure that you don't have any mites present. Okay, so we'll discuss pesticides shortly. Now on to naturally occurring mite predators. So these are predators that may feed on various different groups of mites. Stepharis is a lady beetle that's about 1 to 2 mil. It feeds only on spider mites. Both the adults and the larvae only feed on spider mites, and they can be quite voracious. So if you have these sorts of things present, you want to conserve them if at all possible. This is a delid mite. They are relatively large red mites that are very fast. It has that teardrop shaped. They have relatively long legs that you can't really tell in this picture because it's stuck to a sticky trap, and they're very fast. So some of you may well have seen this sort of thing on a leaf. They can be like bolts of lightning practically, just they're that fast, they go shoo, shoo, all around your plant, and they're very relatively common. Most people have seen these even if they haven't recognized that they're dealing with a predatory mite. There are many other species of predators that you may find in a crop. And in some cases, there are pests that may also feed on pest mites. So for instance, western flower thrips will sometimes feed on two-spotted mites when they are available, but that doesn't mean that you should keep your western flower thrips. I definitely think that it's worthwhile getting rid of those. But there are some species of thrips that are solely predators, and those are worth conserving. So back now to the final stage of the webinar. We'll talk about pesticides and how to incorporate them into a biological control program, and then on to further resources. So biological control is more difficult than just using a pesticide. Hands down, a pesticide, you apply it, make sure you selected the right pesticide, and that's not always straightforward, but you put it on the crop, they normally will die in short order, and then you know whether it's worked, you look afterwards whether it's worked or whether it hasn't. Biological control takes more monitoring. You need to know the type of pest or the type of mite that you're dealing with, type of pest mite you're dealing with, to know which predator to apply. You need to look at the level of pest infestation, whether it's a low, medium, or high. If you're dealing with a medium or high level pest infestation, you need to put the biologicals, you need to delay applying predators until you have a low population. Biological control just doesn't work at high pest pressure, at least not without significant damage occurring. So you need to evaluate if there is a predator that's suitable, if the environmental conditions are suitable for that predator. If it's too hot or too cold, your predator is unlikely to be successful. You need to look at what pesticides have been applied and what pesticides you might have to apply for other reasons in the near future. And if these aren't compatible, then you need to think about whether or not it's worth using predators at that time. If you have a moderate or high level of infestation, then you need to look at applying a pesticide, a suitable pesticide, and know how long that pesticide will have an effect on predators that you release. If it's some pesticides, you can wait just 1 day and then release your predators. Others, you need to wait 3 or 5 days and then release predators, and it will be safe. Otherwise, you may be risking applying a product, a pesticide, applying a predator in a situation where it's doomed to failure. And we don't want that. There's no point in applying a biocontrol agent if you know it's going to die. So to that end, this is a table that is in the pest management plan for mites that we put together a couple of years ago. It is very relevant still, even though it's 2 years old. The only thing that I know of that's changed is this minor use permit 81707. It has a number of different usages which aren't in the current pest management plan. Same active ingredients as we have listed here, but some additional usages which we'll talk about shortly. So these very short residuals will have a low toxicity or a moderate toxicity, and basically after that product dries, you could potentially put on a biocontrol agent and those pesticides would have very limited or basically no effect on them. Whereas some of these other products, you need to wait a few days, or maybe perhaps a week or even 2 weeks for those ones that are highly toxic potentially, before you apply a predator. And then those with a long residual, you need to wait even longer, sometimes even a couple of months. So it's worthwhile talking to your biocontrol agent producer. They have more information about what the length of time you need to wait for each product given particular predators. Different predators have different sensitivities to different pesticides. And while we've only listed insecticides or miticides in this table, fungicides also have a negative effect on predator populations. So talk to them. When in doubt, talk to your biological control agent producer. Build up a relationship with them. They would like to see more contact. They would rather see more contact and have a good result than have less contact and have failure and never have you as a customer again. So, now moving from the biological option to simply using pesticides to manage spider mites in this case. There are a lot of products that are available for use against spider mites. And these are all listed in the pest management plan. Some of these products are compatible with using biological control agents as indicated by the table that we saw previously, but not all of them, particularly your organophosphates, your synthetic pyrethroids, your fipronil, the neonicotinoids. They knock out a lot of predators for a long period of time. So that's okay to use those products, of course. It's just you need to be aware of the effect that they'll have on your predator population. Now, different spider mites, as we talked about earlier, have different levels of pesticide resistance. We have talked about those specifically for two-spotted mite in the two-spotted mite fact sheet. And you have to be aware that sometimes pesticide resistance can occur where it hasn't been published. So you need to keep track of it on your nursery. If you rotate between three or more mode of action groups, you are very unlikely to induce pesticide resistance. And that's why we've listed quite a number of these products so that you can be aware and you can make use of them. For other mite groups, there is a more limited number of products available, and we've summarized them for most of the others. So this is covering flat mites, eriophyid mites, your tarsinevid mites, and other mites, because particularly that permit 81707 has usage of some of these products for mites generally. So potentially if you were exporting and you had a problem with predatory mites, then you could apply those products even to the plants for that purpose. Now, some of these groups, as we've listed down the bottom here, the group 2B is only for tarsinemid mites and eriophyid mites, and group 3B is only for eriophyid mites. You can refer to the pest management plan as well as that permit 81707 in more detail at your leisure. Okay, so we're going to go on to other information resources that are available to you. It is worthwhile building up your own library. Every grower produces a slightly different subset of plants, and there are thousands of plant varieties and species that are being produced across Australia. So take your own pictures of the pests that are pertinent to you, of the damage that they cause pertinent to your plants. And you may also like to get books, reference books that will help you in identifying different pests. It is worthwhile having your pest or your disease identified by a devoted diagnostic laboratory. Don't just rely on the book, because sometimes, the book can't talk to you. You need to make sure that you're getting the right advice for the right pest. And there are a lot of fact sheets out there, whether they're from Australia, like the ones that we'll talk about in a few minutes, or from overseas. So, as we indicated earlier, this current project has many fact sheets and some pest management plans available online. And so what we're going to do is just jump to, sorry about that, pest fact sheets. These are some of the fact sheets. These are all the fact sheets that we have currently available that we've produced under these two projects. So it's becoming quite a substantial list. And as I said earlier, they are designed for the Australian production nursery industry. There are also pest management, a smaller number of pest management plans. These are more detailed than the fact sheets, and they go into specific recommendations about how to deal with particular problems given certain scenarios. You do have to modify them slightly because each nursery is a little bit different, but they are more detailed. There is also a new website. I'll just back up a page here. The Nursery Production FMS, the Farm Management System website, it's relatively new, it's under development. There's a lot of information that's being added to this. If you go to the technical information page, you'll see that there are a lot of different fact sheets available on a variety of different topics. Of course, we're talking about these pests and diseases, and these duplicate the ones that are present in the links we just looked at, but also additional fact sheets. I might just add, going back to the NGIA website, it is worthwhile when you're looking for these pages to type in the search pest management plan or fact sheet, because the link itself is not necessarily intuitive, and it's easier to find them using that search method once you're on the NGIA website than any other method that I've found. Okay, so the last resource that you may or may not know about is this Pest ID tool. It is also designed for the Australian production nursery industry. It is a levy-funded tool. It does have a small subscription, but it is heavily subsidized. So there are a lot of different groups, and the part of the project is adding to this tool. So we'll go to the mites because that's our topic. Oops, we have to log in. And we'll go to browse pest insects and mites. And then it lists a number of different mite species, and then you can click on bee and spider mite, and you get a larger image. So that can help with trying to narrow down what you might be dealing with. Okay, so we're going to pop back to the PowerPoint slideshow before we finish off. Okay, now we talked about sending samples off to a diagnostic laboratory, and that part of this project, the project that I'm involved in, has a diagnostic capacity. So you can send pests or disease-affected plants to us. What we will do is identify that pest or disease and provide recommendations on how to manage it. Lastly, also, please send me your spider mites, and you can email me either through the Grow Help webpage. If you Google Grow Help Australia, it will come up relatively high, if not the first link, and there's more information on how to contact us and submit a sample. So that's it. Thank you for watching this YouTube video of the webinar. If you have any questions, you can contact us at the Grow Help email address, which is there. This is finally a Levy-funded, part of the Levy-funded project, and Hort Innovation Australia funded, as well as the Queensland government funded. Thank you very much. I hope you've enjoyed it. Bye-bye.